Transcription. The first step of gene expression. Transcription is the process by which the genetic information encoded in DNA is copied into a complementary strand of messenger RNA. This process occurs in the nucleus of eukaryotic cells and begins when a specific enzyme called RNA polymerase binds to a region on the DNA known as the promoter, located upstream of the gene to be expressed. Once bound, the DNA double helix unwinds. An RNA polymerase reads the template strand of the DNA. It synthesizes a single-stranded RNA molecule by matching RNA nucleotides with their complementary DNA basis, except that, in RNA, uracil replaces thymine. As the RNA polymerase moves along the DNA, the mRNA strand elongates until it reaches a termination signal, indicating the end of the gene. The mRNA strand then detaches from the DNA, and in eukaryotes, it undergoes several modifications before leaving the nucleus. These include the addition of a 5' cap, a polyetail at the 3' end, and the removal of introns through a process called RNA splicing. The mature mRNA is then transported out of the nucleus into the cytoplasm, where the next phase translation takes place.